first introduce the Asia Center for Health Security for those who have joined us for the first time. The Asia Center for Health Security is a think tank at the NUS, National University of Singapore, Saw Swee Hock School of Public Health. We were launched in April of 2024, and our main work is in the areas of pandemic preparedness, as well as global catastrophic biological risk, synthetic biology, emerging technologies, and biorisk management. My name is Po-Lian Lim. I am the lead for biorisk management at ACHS. My background is as an adult infectious disease doctor, specializing in outbreak preparedness, vaccines, travel medicine. I was director of the high-level isolation unit at the National Center for Infectious Diseases in Singapore, and I currently serve as group director for the communicable disease programs, the outbreak response divisions at the Communicable Disease Agency of Singapore. So glad to have you, and I've been hosting these webinars for the past year. This is our fifth one in our series. I'd also like to give a warm welcome to our ACHS Fellows for 2026. We have a new cohort of Fellows, and would like to warmly welcome them as they join the webinar. So in regards to this topic, this has been a topic that's garnered a lot of interest, looking at AI biodesign tools. RAND Europe has sort of been working on an evidence-based framework to assess biological capabilities and potential misuse of these AI-powered biodesign tools. So the talk by Sana will describe the analytic approach, key findings and insights, and we would like a robust discussion of implications for policy, for responsible innovation, and international coordination in order to make sure that these tools remain safe, secure, and beneficial. So we're delighted to have Dr. Sana Zakaria, Senior Research Leader for Emerging Technology at RAND Europe, to take us through this topic. She leads work at the intersection of biotech, AI, and global security, and advising governments and multilateral bodies on innovation, risk assessment, and governance, as well as technology foresight. Her recent work has involved development of these analyses for the Biological Weapons Convention, using a tech-centric lens on compliance and verification and decision frameworks for global wastewater surveillance. Dr. Zakaria holds a PhD in molecular biology, and she's an advisor for the Science Policy Committee of the UK Royal Society of Biology. So please join me in welcoming Dr. Sana Zakaria. Sana, great to have you. Please go ahead.
It's wonderful to be here to talk about this topic, which has consumed a lot of my time over the last year. So I'd like to talk to all of you about biological design tools and why we chose to focus on this area and how we went about it. One of the first things I want to start with, a lot of you might be aware, but some of you might not have engaged with these tools at all. But a lot of people talk about the dual nature of these AI-enabled biological design tools, or BDTs, from here on. And they are phenomenal tools which are maturing and catalyzing at phenomenal pace. And it's evident in the fact that the Nobel Peace Prize a couple of years ago was awarded to the developers of such a tool. And so BDTs are phenomenal in looking to accelerate drug discovery, vaccine design, and they have really compressed the experimental discovery-based work that we perhaps invested a lot of time in. And, a lot of these tools, they don't obviously work as designed. It's not a straight swap out for the scientific enterprise, but nonetheless, they have provided a lot of interesting avenues for further research and discovery. Now, this obviously transformation or this transformative power, it does come with dual use risks because that very same capability, the very same tools that advance biomedical research can be repurposed for harmful applications. So, for the creation of novel pathogens or to circumvent any existing countermeasures that you might have to evade the immune response that we all possess to be able to fight pathogens. And so this space does require that we remain vigilant and we have some oversight and some mechanism to monitor the trends in this space where we can then sort of think about when is a good time to implement safeguards. What should we be doing? And there really wasn't, when we looked, there really wasn't any evidence base around the capabilities of biological design tools in a structured, comprehensive manner. Whereas for large language models, such as ChatGPT and Claude, there was a lot of evidence base amassing on their performance, their capabilities, what they can and cannot do compared to human performance. The other thing to understand about BDTs is that they provide a really different threat surface to large language models because for a few different reasons. So they direct biological action. So they work on the biological design space. They can manipulate genetic sequences, protein structures with precision, right? So the general purpose LLMs are not quite able to do that in that way. So we're talking about a slightly different space to operate in. They've compressed, like I mentioned, the iteration cycles in biological design, where data and prediction drives workflows and accelerates the discovery and optimization of this biological capability. And this is where, the primary risk is not quite what you would say misinformation or persuasion that might come from LLMs. It's the accelerated development of novel capabilities in the wrong hands. Now, you might say, well, BDTs are really complex to use. So what's the problem? We should just worry about LLMs. But I would say that technical complexity is not the same as safety, because the reason why these tools are, worth oversight is that very few people and labs might be using these tools directly. But for those who want to do harm are unlikely to use these tools directly if they're a novice actor, as you would say. But these tools are connected to general purpose AI for planning and reasoning. Now, they are now embedded in agentic systems that automate a workflow. So you actually don't need to understand biology to a large degree. You don't need to be able to use these tools directly. You can just engage with them through an LLM, through natural language prompts. And that is the reason why we were interested in looking at BDTs. It's not because we are worried that scientists will misuse these tools. It's because we are concerned that what happens when the tools get so mature that they're actually quite easy to misuse, and they are plugged into interfaces that are really easy to use for people who don't understand science. Obviously, in science, we always have insider threat. We always have the science and where science enterprise goes wrong. So all of those things are things that we need to safeguard against. But the main thing I want to kind of put out there is that this kind of integration lowers the misuse barrier. And that's why biological design tools are interesting to look at. Now, what we really wanted to understand is, how do we think this risk scales across the BDT ecosystem? Are there already things that might cover this type of a risk? And so we found that the BDTs, they don't really fit this LLM-centric governance in the way that a lot of the safeguards and safety training mechanisms for LLMs are focused on things like content moderation. So you might try and put something in GPT or Claude or others and say, our policies don't allow us to give you this information. So that's content moderation, content filtering. There's alignment training, where you align the LLM to human-centric values so it doesn't try and do something in a purely logical way, but values-based way instead. There's harm refusals. You can't apply a lot of those things to BDTs because the whole point of using BDTs is that you want to understand what is that one mutation that's going to create a highly virulent pathogen strain, because you want to be able to create vaccines and countermeasures. So harm refusal is basically going to completely undermine the purpose of a BDT. So that really doesn't work in the same way that it might for LLM. The category is also shifting, right? What are BDTs? A lot of BDTs now are actually not niche tools, but they are foundation models, and a lot of general AI capabilities and foundation models that are for biology, their definitions and their boundaries are blurring. And so when you can't really define something, it makes it really harder to then create benchmarks and actually have oversight and do risk assessments, because the boundaries are constantly shifting in this space. But on the flip side of it, if you have really static categories, then as capabilities combine in novel ways, that rigid classification then also doesn't capture the edge cases. So it's a really interesting dilemma space to be in. Now, the other few important points to think about is that BDTs require really modest resources, right? So BDTs can be run on modest compute, and they draw on open biological data sets. That really lowers the financial and technical barrier to entry. Open source models are quite common in BDTs, and I'll talk about what we found in that regard. But once that information is out there, you get irreversible global diffusion of capabilities. Once these tools are out there, and that is the same for LLMs, you can't recall it. It's, you can't restrict it after the fact. It's really hard to do so unless it's a really niche tool buried somewhere in the depths of GitHub. These are globally distributed developments and capabilities. These developments in BDTs are coming from a range of countries, not from a handful of countries, which means that coordination around governance is quite challenging. And like I mentioned, the traditional choke points for LLMs, where you might say, well, we're going to have restrictions like hardware export control restrictions. These don't apply to BDTs because, like I said, they don't use massive compute infrastructure. So that's why I said that they represent a very different threat surface to LLMs. Now, what we wanted to do was develop something that's structured, repeatable for assessing the misuse potential from these tools. And we wanted to present a current kind of landscape assessment of what is state of the art, what is happening. And by no means did we think that our assessment would say, this is the definitive assessment. What we really wanted to do was, because there are so many tools in this ecosystem, almost create a funneling process to say, let's not worry about like the thousands of tools that are out there. Let's help in a very structured way prioritize and funnel this down. So then that delta becomes manageable and help prioritize key tools for deeper assessments that might be more interactive and computationally driven. This is the assessment framework that we developed. And so I'll walk you through all of the steps. But in the first instance, we had to define categories. Like I said, if you just go in and type BDTs, very little will come out because the term itself doesn't hold a lot of meaning. And so we thought about, well, what is it that we want to try and understand, and how do we find out? And what we really wanted to understand is, what is the biological capability and functional capability of these tools? I wasn't really interested, my team, and I should mention, this is the work that we did with Center for Long-Term Resilience. So this is Iran Europe Center for Long-Term Resilience joint endeavor. We really weren't interested in, all of the hype around, oh, these tools will transform life sciences or they are going to destroy everything. So the truth probably lies somewhere in the middle. And so what we really wanted to do was really take an evidence-based approach here. And so what we wanted to understand is, what is the biological capability of these tools? And to understand the biological capability, we created different functional biological domains so that the tools could slot into that. So you have viral vector design, protein engineering, host pathogen interaction prediction. So these are all the biological and functional outputs that these tools produce. And so we found that after a series of iterations, these 8 categories at that time when we did the work seemed really apt and comprehensive and covered all of the range of tools that we were finding in our searches. And these categories are not mutually exclusive. So some of the tools could slot into multiple categories because they do all of that. In fact, Based on what I'm seeing, I find that, we're going to see a divergence in a small set of tools that will be very niche and specific to a single category, and then some tools that are going to get more into that general purpose capability space where they might be able to do a range of all of these things as well at one given time. So the first step was characterization and slotting the tools into these categories. Then we conducted the search around these tools, and we created a range of search terms associated with those 8 categories. And we arrived at 1,100 tools. I'll talk a bit more about details of our search strategy and the shortlisting process. But basically, like I said, we didn't want to analyze every single tool. What we did was we looked deeply at those 1,100 or so tools that were surfaced, and we assessed them for state-of-the-art capabilities. Now, here is also another challenge. Every time people publish a tool, they say this is state-of-the-art. And so what we had to do was come up with a mechanism to understand what is actually state-of-the-art. So when a tool predicts something like entry into a cell, how good is it? Is there in vitro validation of that capability? And because there are no benchmarks, you can't just simply slot it against a benchmark and say, this is how these different tools perform against the same task. We had to make our own call and read the scientific papers and assess how well are these things performing. And so that's how we arrived at the 57 tools across those 8 categories. And then we did 2 main types of assessments. One was the landscape assessment, where we were looking at across all of the range of tools, what's the time of release? We did something called risk chain mapping, which I'll talk about in a moment. We looked at things like potential for change, how likely are these capabilities to change, or how likely is there going to be wider ecosystem change, such as a massive inject of cash or infrastructure investment that might catalyze developments in this space, because we wanted to understand what is happening in the wider ecosystem surrounding these tools as well. And then the tool level assessment was really looking at what's the misuse relevant capability of these tools and what is its maturity and availability. I'll walk through these concepts in a little bit. So this is the search and shortlisting methodology that we deployed. We did a lit search, we had expert crowdsourcing, and we did some targeted searches through a snowballing approach, looking at papers that mentioned other papers. And we, like I said, we arrived at unique tools. There was actually multiple steps to shortlisting. So first, we shortlisted tools with potential frontier capabilities. And then from that, we arrived at what we call a minimal frontier set. So this is where we chose the tools that could conduct a diversity of tasks within a category, variety of tool types and computational architecture, transformer models, graph neural network, all of the different kind of machine learning capabilities built into the tools, and then different misuse relevant capabilities as well. This is what the risk chain mapping that I was talking about. So to understand the landscape of tools and kind of their maturity, what we did was we mapped them against what we call the biological weapons design chain. And it's your typical DBTL cycle, so design, build, test, learn. Within the design stage, you think about a couple of things. You think about what's the agent, what's the pathogen, and what's the delivery mechanism. And then you kind of, go through that cycle, and then you weaponize and deploy. What we wanted to understand was, okay, based on biological function, which of these steps could the tools help with? Now, because of information hazards and doing this outside of government, we realized that it might not be a great idea to map it at the tool level at this stage, unless you were doing it in a classified setting. So we decided to do it at the category level. So protein engineering as a category with all of the tools, what steps could it enable? And so we did that mapping, and a range of tools kind of mapped all of these different steps. And this is published in our paper, so you can go into the details at that point. But the point of doing this exercise was really to see which steps of this pathway were enabled by which type of tool categories. And again, I think what we are seeing here, a very maybe preliminary trend, is that a single category can now straddle multiple steps. So it's not that you need to piece together different types of categories and capabilities to achieve all of the steps, but actually what we are seeing is that one single category or one single type of tool over time may be able to do all of these things. We also looked at, like I said, maturity and availability of the tools, because that's an important factor, right? Because if you have a tool that's really embryonic, really nascent, or it's buried in the depths somewhere that nobody can access, that kind of reduces its misuse potential. So we wanted to take that into account. And so this was a 5-point rubric that we developed around maturity of the science and technology, the demand signal, political, moral, ethical barriers to the technology. Are there any kind of barriers foreseen where people might not engage with it because of some kind of a taboo or other kind of challenges, funding resources being put into the tool, and technical expertise required and availability to the public. And that kind of was a 5-point, so altogether 25 points that were possible from this. And then we kind of had a weighting criteria. And then we came up with this overall maturity and availability score. And the higher the score, that means those tools are mature and highly available as well. This is explaining the other main crux of the misuse assessment. This was the misuse relevant capability. Now, I'm not going into the details of that rubric because there are 8 of them. So for every 8 single biological category, we had 8 misuse rubrics. In each misuse rubric, we had 2 to 3 misuse scenarios. So that's a lot of information to go through. But basically, when we had a tool, let's say AlphaFold in the protein engineering category, we used the rubric for that category and said, here are potential misuse pathways. And then these are the technical capabilities that this tool needs to enable. And in those technical capabilities, we had different scales, from low misuse to medium to high to critical. And so for critical, there were certain parameters that the tool had to enable around novel design, something that hasn't been published before, novel capability, novel mutation, immune evasion, a range of different things, depending on which biological category the tool belonged to. And if it fulfills all of the criteria, as we can see in the publication, then that means there is a critical misuse level ascribed to that tool. We did that 2-level assessment. Of course, we haven't published it. We published our methodology, but the analysis of the 2-level is only for government use for obvious reasons. Now, this is the rubric. This is the composite score rubric. So based on a single tool's ability to have a certain amount of misuse associated with it for a specific biological category, plus its maturity and availability, which I just went through, that combination is what gave us this rating of red, amber, green. And so this is kind of the scoring rubric that we came up with. Anything that's high or critical risk would go into the red category. But anything that is high could also go into the amber category if it's really not mature or not available. So amber category is also a really important category, I just wanted to say. And you could also have tools in there that have medium misuse relevant capability. But again, their maturity and availability is quite low. So that's kind of the range. Like I said, this was a judgment call, which is why we did the 2-level assessment to really unpack every single tool for the government, for policymakers to kind of be aware of this operational space. Now, some interesting findings were that, all of the tool categories are growing significantly. So we did our search from 2019 to 2025 to early 2025. That was the cutoff for our analysis. And you can see on the graph, there's just phenomenal growth in that space in terms of the number of tools across all of the different categories that have grown since then. What we also saw was that a lot of the acceleration of frontier capabilities in biodesign tools emerged in the last 18 months at the time of research. So state-of-the-art tools that we had selected in our analysis, the 57 tools, you can see on the pink where it says yes, these were the years that they were released in. So quite a bit of a jump in 2023 and 2024. The reason why it's low for 2025, like I mentioned, our cutoff was at the start of the year. So this is obviously, this bar is not representative for the full year. But I imagine the trend might remain. This is the other point I made, that these tools, the 1,100 or so tools that we found, they were coming from everywhere, all across the globe, so across 76 countries. So it's not that, they are developed in a handful of nations. And so that has implications for governance. So we can talk about governance in the, US context, the UK context, but that is actually not going to create global safety and norms in the way that we would wish to. So global international coordination would be key. The frontier tools, the 57 that we singled out for assessment, came from 24 different countries. So again, it's not a small amount. It's quite a significant spread of countries that were involved across multiple academic collaborators. I'm obviously not going to present a two-level assessment, but I wanted to share, again, additional insights on the number of tools that we had across each category. So here are the biological categories that I shared earlier. And if you see in the viral vector design, there were 6 tools that were at the frontier that we analyzed. 3 of them were green and 3 of them were red, which means that there is a very high chance of misuse, basically, and that we recommend immediate action. So that's what these RAG ratings signify. Red is basically very high misuse potential. Take action now. And there's a range of recommended actions that we put forward. Amber means assess on a case-by-case basis, but again, a very high chance of misuse. And green means wait and see, keep an eye on these things. So you can see protein engineering, very, very advanced compared to some of the other categories. And we had 8 tools that were red on there. So it's an interesting analysis to kind of break it down in this way. And you can see quite a lot of ambers here across genetic modification, experimental design, simulation, and automation workflows as well. And I imagine this number will have gone up since we conducted our analysis as well. And even in that, we had one red tool or red workflow. So all across the board, we had about, I would say about 25% of the tools were scored red, which means that 25% of the tools at that frontier really require immediate action. And the real number might be higher. Obviously, our methodology is not perfect. We are humans, we make mistakes. So in analyzing those 1,100 or so, it might be that we didn't pick up everything. It might be that in our assessment process, instead of 57 tools at the frontier, there might be 65. So it's not a foolproof method, but it's a proof of principle to kind of show that you can do these evidence-driven assessments, and actually doing so in a structured way could be beneficial to get to grips with what is the capability and maturity of these tools and the ecosystem, and when should we be concerned. We also looked at a comparison of misuse capability against maturity and availability, just to see whether there was any correlation between something. If something was low misuse, was it also less available? If something was critical misuse, was it more available? But actually we didn't see a correlation at all in this relationship. So that was just an interesting thing that we found. And like I mentioned, so yeah, we had about 23% of the tools that were red, so recommended action, and then 26% amber, where we said consider action, and then 50% where we said no immediate action required, but wait and watch and see. Now, the other thing, I guess the more concerning thing that we found that we weren't expecting is that over 60% of these red tools were completely open source. That means anybody can just download all of that. You don't need to use an API. You can use it locally on your computer, and fine tune the tools and use them and train them on a different data set if you like, to kind of really catalyze its capabilities further. Which also means that a lot of these amber tools, the reason why they're amber is because we found that maybe they weren't trained on the data set that would make them more dangerous. Very easily, somebody could take these amber tools and make them red by training it on a different type of data set that's more relevant to humans, for instance. Um, Recently, just wanted to call two things into action. So, these are not hypothetical things. Again, people might sort of say, well, still BDTs can't do so many things, but a lot of them can. And these are two examples of recent kind of, I would say milestone moments where you had a study done by a range of researchers, Microsoft, IBIS, other collaborators who used a series of BDTs that generated sequences for a pathogen. And so they put them through something called a synthesis screening tool. So this is where you screen for sequences, because when you want to conduct an experiment in a lab, you order the sequence for something, a virus. You don't just make it from scratch. And a company or whoever is giving you the sequence might screen for it, say, let me just check what is it that I'm sending out. And so what this experiment did was that they created sequences. Let's say they didn't create smallpox, but they created sequences for virulent pathogens, but they prompted the BDTs to create sequences that would create the same function as the pathogen, but the sequence similarity wasn't there. And the reason why that's important is because the screening tools that are based on sequence alignment didn't pick up any of these AI developed, AI predicted sequences. So if you do that, that means you can effectively get your hands on dangerous sequences that don't resemble the sequences that we know to be looking out for, but can function perhaps in the same way. Now, obviously, this needs experimental validation in the lab to see whether any of the predicted sequences would actually function in that way. So that perhaps might be the next iteration of this. But this was a really interesting experiment to say, even our defenses, how resilient are they to AI advancements and capability uplifts? The second one was this AI design. So EVO1 and EVO2 was used to design viral genomes. So, and they were successfully synthesized into a bacteriophage. So bacteriophages are viruses that gobble up bacteria and fight bacteria. And so they were able to use these generative models and create something that was functional. And actually, these bacteriophages could infect and, kill E. coli bacteria in the lab, and it performed better than wild-type bacteriophages. So these tools can produce functional outputs is the point I want to make. These are the range of recommendations that we came up with around, obviously we break them down a lot more. These are some high-level recommendations around international governance coordination, investing in mitigations and technical safeguards that are more relevant to BDTs rather than LLMs, and also conducting more regular assessments of this space to monitor the trends and capability uplifts. And so I'm pleased to say that as a result of that, we have been funded to, in fact, set up something called a Risk Index Observatory. So this is something that will consume a lot of my time over this year, is to establish this mechanism, a repeatable mechanism for monitoring and assessing these BDTs. And we're going to be undertaking another process for doing tool identification, doing the risk assessment, doing more analytics on this and creating dashboards and flags for assessment, but also automation, seeing if we can kind of create an always-on capability where we have flags for any new tools that emerge that are worth a deeper look on its misuse potential. And we will also be creating something called a Rio calculator, which is where developers can put their own tool through the rubric in a very easy way and actually see whether, how misused relevant is their tool. Because we know that the tool developers and the scientific enterprise, they don't think about these risks, right? Because traditionally, why would they? They want to just create the next transformation in science. So this calculator will hopefully help with that kind of frame and shift in thinking as well, to make sure that when we do go out and deploy these tools, we do it in a safe manner. And I believe that's the end of my presentation.
Thank you very much, Sana. This is really fascinating.